inside nearly every cell in your body. There's a 3 billion letter instruction manual that dictates how that cell should behave. This manual is called DNA, and it turns out that this manual is not only inside you, but in almost every living thing on Earth. Here's a fun fact. You have about 2 meters of DNA packed into every single one of your cells. But what actually is DNA? Well, it's a molecule. As you can see, A always pairs with T, and C always pairs with G. We call these base pairs complementary base pairs, because these base pairs code for amino acids, which are the building blocks of proteins. First, we divide the gene into groups of 3 base pairs, starting from a specific place called the start codon. Remember, each group of 3 is called a codon. By using certain enzymes, we can cut DNA at specific sequences. In today's world, it's impossible to succeed without understanding STEM concepts like DNA. You need them for everyday things like understanding climate change or pandemics. But you also need them for success in the workforce. The demand for workers in STEM fields is growing faster than ever before. And then we can insert a new piece of DNA with a different sequence, and the cell will incorporate the new DNA into its own genome. Here we have the original DNA molecule in blue and red. Then we unzip the molecule and separate the 2 strands. Let's take the gene for eye color. The gene has 2 different alleles, which are different versions of the same gene. The B allele codes for brown eyes, and the B allele codes for blue eyes. Let's say a person has 1 brown eye allele and 1 blue eye allele. You know how it stores genetic information, how it replicates, how it can mutate, and how it can be packaged into chromosomes. DNA is truly the blueprint of life, and understanding how it works is crucial for understanding biology. Since the B allele is dominant, the person will have brown eyes. If you did enjoy this video, please hit the like button and subscribe if you haven't already. And as always, thank you for watching, and I'll see you in the next one. However, if the person has 2 blue eye alleles, then the person will have blue eyes. Now, let's imagine that the B allele mutates and becomes non-functional. This means that the person will have 1 functional B allele and 1 non-functional B allele. Since the non-functional allele is recessive, the person will still have brown eyes. But now, the non-functional allele can spread throughout the population, leading to more people having blue eyes. Each strand acts as a template for making a new complementary strand. The green strand is the new strand that is being synthesized, and the yellow strand is the new complementary strand. And voila! We have 2 identical DNA molecules, each with 1 original strand and 1 new strand. Instead of memorizing formulas and equations, Brilliant allows you to learn by doing. Mutations are permanent changes in the DNA sequence. They can occur spontaneously during DNA replication, or they can be caused by exposure to things like radiation or certain chemicals. Most mutations are harmful, but some can be beneficial. Beneficial mutations are the raw material for natural selection. They introduce new traits into a population, which can lead to the evolution of new species. Chromosomes are simply condensed structures of DNA and proteins. You inherit half of your chromosomes from your mother and half from your father. Humans typically have 46 chromosomes in every cell, but the number of chromosomes varies among species. For example, dogs typically have 78 chromosomes. There are over 60 interactive courses on Brilliant covering topics from foundational math to advanced computer science. One course that I really enjoyed was their scientific thinking course, which teaches you how to think like a scientist and engineer. It covers problem solving, data analysis, and experimental design. It's such a fun course. For example, scientists have used genetic engineering to create golden rice, which has beta-carotene, which is a precursor to vitamin A that can help prevent vitamin A deficiencies. Another example is genetically modified mosquitoes that resist malaria. By using genetic engineering, we can cure genetic diseases, improve crops, and even fight the climate crisis. Now, each codon corresponds to a specific amino acid. We can use this chart to translate the codons on the DNA to the correct amino acids. For example, the first codon in this gene is August, and according to our chart, August codes for methionine. The next codon is GCU, which codes for alanine. And the final codon is UA, which is a stop codon. That means when a cell divides, it can pass down an exact copy of its DNA to its daughter cells. And this is possible because the two strands of the DNA molecule are complementary. Remember, A pairs with T and C pairs with G. Because of this, if we take apart the two strands of the DNA double helix, each strand can act as a template to make a new complementary strand. So when we replicate DNA, we end up with two identical DNA molecules, each with one original strand and one newly synthesized strand. This process is called semi-conservative replication because half of the DNA molecule is conserved in each of the daughter molecules. Stop codons tell the cell to stop adding amino acids. So the resulting protein from this gene is methionine, alanine. Let's explore how this works. Look at this sequence of base pairs along your DNA. This is a gene. A gene is simply a segment of DNA that contains the instructions for building one protein. But notice that there are gaps between the instructions. You see, the alphabet for DNA is only for letters, but the alphabet for amino acids, which are the building blocks of proteins, is 20 letters long. So how does a 4-letter DNA alphabet code for a 20-letter amino acid alphabet? Well, the answer is that each amino acid is coded for by a 3-letter combination of DNA. These pairs form the rungs of the ladder, and then the sides of the ladder are made up of alternating sugar and phosphate molecules. In reality, the ladder twists and coils in on itself to form a double helix shape. It looks like this. They're responsible for everything that happens in your body, from catalyzing metabolic reactions to transporting molecules across cell membranes to fighting off pathogens and much more. And all of this happens because of the sequence of amino acids in the proteins. So by changing the sequence of amino acids, you can drastically change how your body functions. And mutations in your genes can lead to changes in the amino acid sequence, which is the root cause of many diseases. And this is why we often draw DNA as a twisted ladder. But if you were to straighten it out, it would be a long, flat ribbon. Molecules are just different combinations of atoms bonded together. And if you remember back to high school chemistry, you'll recall that the basic building block of all molecules is a chemical called a nucleotide. Just like how Legos can be put together in many different ways to make different molecules, nucleotides can also be put together in many different ways to make different molecules. And DNA is one such molecule. So let's learn how DNA works, the blueprint of life. First, let's zoom into one of your cells and look at the nucleus. That's where we'll find your DNA. DNA is made up of 4 different types of nucleotides, And we represent these nucleotides with the letters A, T, C, and G. Now, just as the order of letters in a word determines what that word means, the order of these 4 letters along your DNA strand is what determines how your body functions. Each individual unit along the DNA strand is called a base pair. So we read the DNA sequence by reading the base pairs. Now, here's another fun fact. If you were to read the entire human genome, which is the entire set of DNA instructions for the human body from start to finish, it would take over 3 minutes to read the entire thing aloud. Now, these nucleotides come together in very specific ways. Now, the reason why we care so much about the exact sequence of these base pairs is, let's take a closer look at this gene and decipher what it codes for. So now, we know that this gene codes for the protein methionine alanine. But remember, proteins are what carry out the instructions in your cells. But sometimes we want to make changes to the DNA sequence intentionally. This is where genetic engineering comes in. But here's the key point. All of this is possible because we first need to understand how DNA works. And Brilliant is an amazing tool that makes learning STEM concepts interactive and engaging. Thanks for watching this video, and thanks to Brilliant for sponsoring it. Now, let's get back to the video. Another cool thing about DNA is that it can replicate itself. We can actually visualize this process by using different colors to represent the 2 parental DNA strands. Another important property of DNA is that it can mutate. Here is an example of how a mutation can affect the amino acid sequence of a protein and ultimately the trait of an organism. Mutations are a powerful force of nature, and understanding how they work is crucial for understanding evolution. Another important property of DNA is that it can be packaged into chromosomes. All right, so now you know how DNA works. Hopefully you enjoyed learning about DNA, and hopefully this video helped you get an A in your class or on your exam.